Hello, my name is Sara Samir Alsharif. Today I will talk about my PhD work, and it's about novel fluorescent platinum derivatives and their delivery by fusogenic liposomes as nanocarrier into cancer cells. I'm doing my work in Institute for Biological Information Processes, IBI-2. My work aims to, first, development of highly efficient cancer therapy and reduce the side effects. In addition to that, development of novel platinum chemotherapeutics and the delivery of those novel platinum chemotherapeutics by fusogenic liposomes. The human health is one of the most important area to focus in. So, as all of us know, there is many diseases can affect the human health. And to treat it, we need to deliver the appropriate drug to the target cells. But it may be complicated than you think because of the side effects, the degradation, and the unable to reach the target. So we need a helper to solve this problem, and it will be the drug carrier. There are many drug carriers can be used in pharmaceutical to deliver the drug, and the one we use in our work is the liposomes. The liposomes are considered to be non-biodegradable, non-toxic biodegradable carriers. They are carriers can deliver both types of a drug, the hydrophilic and hydrophobic, and also they cannot be administered by various routes. The word liposome, it's derived from two Greek words. The first one is lipo, that means fat, and soma, that means body. And it's named because of its composition, which is primarily of phospholipids. As we see here in this structure, that the phospholipids consist of the hydrophilic head, water-loving group, and the hydrophobic tail, water-fearing group. And the number of phospholipids together form phospholipids bilayer. A liposome is a spherical microscopic vessels that can consist of at least one phospholipid bilayer, and their size can be ranged from 50 nanometers to several micrometers. There are different pathways can the liposomes taken by the cells. One of those pathways is the fusion. In this pathway, the fusogenic liposomes will fuse with the cellular membrane that lead to release immediate drug release and increase the uptake efficiency and decrease the degradation. Another pathway can used here as the endocytosis. The endocytosis pathway helps make the endocytotic liposomes to surround by a part of the cellular membrane that will degrade later on by lysosomes, and it will slow the uptake efficiency and reduce the release. Most of nanoparticles taken by cells by endocytosis. So in our work, we will use those endocytotic liposomes as our control. The liposomes. The main composition of the liposomes is the neutral lipids, the cationic lipids, and the aromatic group, which is usually a di. In the fusogenic liposomes we prepared, we have the DOBE as a neutral lipid. DOTAP and DOTMA will be our cationic lipids. They are a little bit different in their structure, in which DOTMA consists of ether bond, while DOTAP consists of ester bond. The ester bond is easier to be degraded by the cells that make it less toxic, but the DOTMA is more fusogenic. And we have the di-R as a di. The interaction between those three components will lead to a red fluorescent signal. So we have a fusogenic liposomes with DOTAP and the highly fusogenic liposomes with DOTMA. The control here, which is the endocytotic liposomes, consists of the DOBC as a neutral lipid, the DOTAP as a cationic lipids, and the di-R as a di. The preparation of liposomes. First, we dissolve the desired amount of those lipids into DMSO instead of a chloroform. After that, dry it until this thin film produced. Disperse this film in HEPA's buffer and vortex for 1 to 2 minutes to produce multilaminar liposomes. After that, sonicate for 20 minutes at 4 Celsius to produce unilaminar liposomes, which can be stored for further use. As I said before, that we use the DMSO instead of a chloroform because our derivatives that we will use later are dissolved in the DMSO, not in chloroform, neither in water. So we need to test the appropriate amount of DMSO needed to dissolve the whole lipid. So we take 10 microliter, 20 microliter, 50 microliter, and 100 microliter. And we found that with 10 microliter and 20 microliter, it wasn't enough to dissolve the whole lipid. And 100 microliter, it was enough, but it was a little bit toxic for the cells. In the case of 50 microliter, it was enough and didn't affect the cells. So we will choose the 50 microliter. The time needed for DMSO to be evaporated, we test several time points. We evaporate for 24 hours, 48 hours, and 72 hours in the executor, and we find that after 72 hours, the total volume of DMSO was evaporated. As an additional step, we make freezing and thawing for 5 times for the liposomes before using to control the size. Okay. Here we can see a microscopic image for mammalian cells that treated with endocytotic liposomes, and also use NucBlue to stain the nucleus, which will help to visualize the cells under the microscope. And as we see here, there isn't any red fluorescent signal. That means those liposomes cannot interrupt the cells. On the other hand, we treat the same cell type with the highly fusogenic liposomes with DOTMA, and here we can see nice fusion rise fluorescent red signal and also a blue signal from NucBlue. And that's what I talk about, that it was a little bit toxic, and we can compare this from the number of the cells between the figures, and we get the highest diffusion efficiency when we dissolve the lipid in 50 microliter of DMSO, and it was 75%. After that, we start with the characterization of the liposomes to determine the size distribution and the zeta potential, and here we use dynamic electrophoretic light scattering. We're using dynamic light scattering to determine the size distribution before and after many cycles of freezing and thawing. And as we see here for the 3 samples with fusogenic liposomes with DOTMA and the endocytotic liposomes with DOTAP, that the size already reduced until we reach almost 100 nanometer. And this is very important for us, especially for an in vivo experiment, because the cells usually uptake those small particles and exclude the large particles. And for the zeta potential, we use electrophoretic light scattering, and we see that with and without the freezing, there is almost no difference between the charge. As a conclusion for liposomes preparation, we developed a preparation method for fusogenic liposomes, determination the amount of DMSO needed to dissolve the lipid and the time needed to evaporate, additional homogenization step, freezing and thawing to control the size. Cisplatin derivatives. Cisplatin is a chemotherapy medication used against different types of cancers like lung cancer, brain cancer, breast cancer, and ovarian cancer. And the treatment usually given as intravenous injection. There are many side effects for the cisplatin derivatives, like ototoxicity, neurotoxicity, and the kidney damage. And the mechanism of how this cisplatin works with the cells, it by cross-linking of the DNA, result in reduced cell proliferation, that may reduce the cell growth and dividing. The lipophilic cisplatin derivatives. Now here we can see the R, which is the sided chain. This R can be varied. My supervisor from the Palestinian side, Professor Youssef, synthesized many compounds with different sided chains, and we can name them AQ2, AQ3 until AQ18. It's not allowed to me to show them because he applies for a patent, so we will name them like this. And those non-lipophilic, they will be our control, and we will try to incorporate them into the liposomes. And Professor Youssef additionally will form more lipophilic liposomes by forming lipid-like chain. This long chain, it's lipid-like chain coupled to his cisplatin derivatives. But unfortunately, because of the situation right now of COVID-19, we have some laboratory work problems, and it's not allowed to enter the university in Palestine. Okay. Now I will talk about the cancer cells treated using those non-lipophilic platinum derivatives incorporated into fusogenic liposomes. We treat cancer cells with different derivatives, and we measure the proliferation percentage. And as we see here, that the proliferation percentage was decreased when we treat the cells with AQ3 and AQ4 compared to the control and other compounds in both with a carrier, which is our fusogenic liposomes, or without a carrier as a free drug in BBS. In addition to proliferation effect, we have toxic effect, and as we see here also, the number of the dead cells increase when we treat them with AQ3 and AQ4. We can conclude that the AQ3 and AQ4 are more efficient than others to treat those cancer cells, but we start with AQ4 because it's a fluorescent compound and it can be detected under the microscope easily. Here we have some microscopic images of cancer cells treated with AQ4 at different concentration incorporated into fusogenic liposomes DOTMA. And we can see from day one that all the cells look healthy and we have a nice fusion. In contrast, at day 6, with lower concentration of AQ4 incorporated with the fusogenic liposomes DOTMA, we can see that the cells are still healthy, but the dye are degraded, and with a higher concentration of AQ4 incorporated into fusogenic liposomes DOTMA, we can see that there is almost no cells because there is a problem in the proliferation and there is a toxic effect, as I said before, because of the high concentration used here. Quantify the effect of AQ4 by viability assay in cancer cells. Here we use the cancer cells and treat them with different AQ4 derivatives with different cisplatin derivatives. The one we use as AQ4 incorporated into fusogenic liposomes or as a free drug in BBS at different concentration. Also, we have cells treated only with the fusogenic liposomes with DOTMA alone without any cisplatin derivatives, and the positive control here we have is the cells without any treatment. And we can see from day one that the cells, the absorbance of the cells treated with the fusogenic liposomes DOTMA is a little bit high. That means that those fusogenic liposomes are not toxic for the cells. On the other hand, if we compare between 2.5 AQ4 incorporated into fusogenic liposomes DOTMA and in BBS as a free drug, we can detect that the absorbance for the cells in a free drug is less than the absorbance of the cells treated with the AQ4 incorporated into fusogenic liposomes, and also if we use the concentration 10. But at day 4, we can see that the absorbance for the cells treated with the empty liposomes, the fusogenic liposomes DOTMA alone, was a little bit lower than day 1, and also the absorbance for the cells treated with the AQ4 as a free drug at different concentration was a little bit higher than the absorbance of the cells treated with the AQ4 in different concentration incorporated into fusogenic liposomes. That means that the AQ4 is more efficient upon delivery by the fusogenic liposomes than in BBS. As a conclusion, here we development of liposomes preparation method in cooperation of these AQ4 cisplatinum derivatives in fusogenic liposomes, and AQ4 is more affected upon delivery by fusogenic liposomes compared to free drug. Our experimental outlook is synthesis of more lipophilic cisplatin derivatives, incorporation it into fusogenic liposomes, and analysis the drug effects on cancer cells. I would like to thank Palestinian German Science Bridge to give us this opportunity, Institute for Biological Information Processes, the head of our institute, Professor Rodolph Merkel. Many thanks to my supervisor from the German side, Dr. Agnes Schizar, and many thanks to my supervisor from the Palestinian side, Dr. Youssef Najajra, and also to Tabia Wittenhofen. Thank you all for your support and help, and thank you all for your listening.